It is now my pleasure to introduce Dr. Andrew Oberst, Assistant Professor in the Department of Immunology at the University of Washington in Seattle. Dr. Oberst graduated from Amherst College in 2001 and pursued his graduate studies in Europe in a collaborative program between the universities of Rome and Paris. He received his doctorate in 2006, then completed postdoctoral training at St. Jude Children's Research Hospital in Douglas Green's lab in Memphis, Tennessee. He then joined the Department of Immunology as an assistant professor in 2012. Dr. Oberst has used Essen BioSciences IncuSite technology in various laboratories for over 6 years to execute live cell phenotypic studies on cell death. He will be speaking with us today about necroptotic cell death, and his talk is entitled Going Out with a Bang, the Pathways of Necroptotic Cell Death.
All right, well, I'd like to start by thanking Essen for giving me the opportunity to give this webinar. I'm Andrew Oberst. I'm an assistant professor at the University of Washington in Seattle. This is actually a photo of me in the mountains near Seattle. It's a great place to live. And I'm going to talk today about cell death. And I'm going to start by sort of presenting the classical view of cell death. This is how we used to think cell death worked. And the way that we used to think cell death worked was that it was a dichotomy, that cells could die in one of two ways. They could either die by this passive, unprogrammed process of necrosis, or they could die by this programmed form of cellular suicide called apoptosis. And necrosis we think of as the unordered killing of cells by an overwhelming chemical or environmental insult. So think things like pouring bleach into your cells in tissue culture, freeze-thaw, toxins, things of this nature. They can cause the swelling and rupture of cells, and when this happens in vivo, we think of it as being inflammatory because it releases the contents of cells, releases lots of reactive molecules that lead to an inflammatory and tissue damage response. On the other side of the coin, we have apoptosis. Apoptosis is an active process of cellular suicide. It's triggered in response to many different types of specific signals, and I'll talk about a few of those in a moment. And apoptosis is really defined by the activation of this family of proteases called the caspases. And this is one of the caspases. Notice that that this caspase has this dimeric structure. This is an important facet of caspase activity. And the caspase proteases cleave thousands of substrates within cells as they undergo apoptosis, and this leads to this distinctive apoptotic morphology that I'll show you in a moment. And apoptosis as a phenomenon is required for normal development, immunity, tumor suppression. It's a very normal part of organismal function. Billions of cells die in our bodies every day by apoptosis. And apoptosis involves distinct hallmarks that are a result of caspase activation, membrane blebbing and nuclear condensation. And when this happens in vivo, this is followed by very rapid recognition and phagocytosis by immune cells. And so this is a depiction of a cell in the final stages of apoptosis being recognized and phagocytosed by a responding immune cell. And because of this, apoptosis is generally thought of as a tidy and non-inflammatory form of cell death. Remember that billions of cells are dying by apoptosis in our bodies each day, and that this generally doesn't cause significant inflammatory responses. So, as I said, this is sort of the old way of thinking of cell death, and I call it the old way because we now understand that, in fact, there are other ways for cells to die. There are other forms of programmed cell suicide that can take place. Two of them that we're interested in studying in the lab are the phenomenon of pyroptosis, which I'm not really going to talk about today, but which is another form of caspase-dependent cell death. And then this other phenomenon called necroptosis, which is what I'll focus a lot of my talk on today. And unlike apoptosis, which is this sort of normal physiological phenomenon, these forms of cell death are generally associated with stress and with infection. And also distinct from apoptosis, they have these very clear and different morphologies that cells take on as they die. And so you can see here's an apoptotic cell. You can see this sort of shrinking and blebbing, consistent with what I've already told you and what I'll show you in a movie in a moment. And when cells undergo pyroptosis or necroptosis, these are primary bone marrow-derived macrophages, you can see that they swell and they actually rupture. And you can tell that they've ruptured because these cells have all turned green. In these movies, we've put a cell-impermeable nuclear dye in the media, and so when the cells lose membrane integrity, they'll turn green. These are all images taken using our IncuCyte Zoom imager. And one of the things that we really love about the IncuCyte for these sorts of assays is that it allows us to not only quantify cell death, but actually to see how the cells are dying. So we get both quantification and a measure of morphology. So I'm going to talk initially just a little bit about some of the apoptotic pathways, and then tell you how these pathways interface with the necroptotic pathways, the pathways that trigger necroptosis in cells. So the extrinsic pathway of apoptosis, it's so named because it's apoptosis in response to signals that come from outside of a cell. These signals can bind to receptors, notably the death receptor such as FAST, the trail receptor, and the TNF receptor. And this leads to the activation of a caspase, one of these proteases that I've already mentioned to you, called caspase-8. This is going to be a key player in the rest of the talk. And caspase-8 activation can then lead to apoptotic cell death, either via direct activation of downstream effector caspases, or in many cell types via the engagement of this other branch of the apoptotic pathway, the mitochondrial pathway of apoptosis, which leads to the release of factors from the mitochondria and formation of these secondary complexes, which then leads to execution or caspase activation and apoptosis. So let's just take a look at this. Here are some HeLa cells that are going to undergo apoptosis. Their nuclei are labeled by a histone GFP. And you can see this very distinctive shrinking and blebbing associated with this form of death. When this happens in vivo, these cells would be recognized and phagocytosed fairly early on in this movie. Subsequently, when this happens in vitro, we can observe this what we call secondary necrosis, which is the rupture of the apoptotic cell membrane that happens later and leads to the release of cellular contents. So one of the ways that apoptosis has been studied over the years is, of course, by making knockout mice that lack key members of the apoptotic pathway. And, we think of apoptosis as a form of cell death that eliminates superfluous or damaged cells. And so you would expect that if you knocked out things involved in inducing apoptosis, the phenotypes of the mice that you would get would be associated with an overabundance of cells, right? You take out the cell death pathway, and you have too many cells left behind. And for the most part, that's in fact what you see. So when you knock out some of these apoptosis-inducing receptors, you get the phenotype of excess immune cells and autoimmunity, so you have too many cells. Knock out some of the pro-apoptotic BCL2 family members, you again can get autoimmunity. In some cases, you get mice that are tumor-prone. In some cases, you can actually get mice that have webbed feet because during development, the interdigital space of the mouse's paw and of our hands are sculpted by apoptotic cell death. So if you remove apoptosis, you actually can get webbed-footed mice. And if you take out some of these downstream players in apoptosis, you can get this exencephaly phenotype in which the brain of the mouse actually protrudes from the skull. This, of course, is embryonically lethal, but this is because of a defect in neuronal apoptosis. These mice have too many neurons, and their brains simply can't fit in their heads. So in all of these cases, knocking out cell death leads to an overabundance of cells. But there's an interesting exception to this rule, and this is the case of caspase-8. So remember, caspase-8 is this enzyme that's directly activated by these receptors. And when you knock out caspase-8, the phenotype that you get is embryonic lethality at day E11, and this is associated with a failure of normal vascularization. So you can see here's a wild type, or actually a caspase-8 heterozygous mouse. You can see this clear vascularization of the yolk sac and of the developing embryo. Caspase-8 knockouts have a defect in this vascularization. And furthermore, conditional deletion of caspase-8 in lymphocytes actually leads to a defect in lymphocyte proliferation and accumulation. So you end up with a mouse whose T cells can't accumulate in response to an antigen challenge. So unlike most members of the apoptotic pathway, caspase-8 seems to be required for normal development and lymphocyte activation. And we wanted to ask the question of why this is. This is work that I did during my postdoc. And the answer to this, I'll cut a long story short, is that caspase-8 is actually required to suppress another cell suicide pathway, necroptosis. And so this is a depiction of what happens in a cell when caspase-8 is absent. Receptor activation can lead to the activation of this kinase RIP1. This will be an important player later in the talk, so keep RIP1 in mind. RIP1 can then translocate into the secondary complex in the cytosol, where when caspase-8 is absent, RIP1 can interact with another kinase called RIP3, and this activation of RIP3 by RIP1 leads to the activation of RIP3 and in turn to necroptotic cell death. So this other form of cell suicide distinct from apoptosis, which is in fact not only distinct from apoptosis but is actually suppressed by key apoptotic player caspase-8. So the caspase-8 knockout mice die because they have overabundant activation of RIP3, and we showed that the double knockout of caspase-8 and RIP3 is now normal and viable. So here I just want to show you an image of a cell undergoing necroptosis so that you can appreciate how distinct this alternate form of cell death is from apoptosis. You'll see that these cells, as they die, rather than shrinking and blebbing, actually swell. They round up like little balloons, and then they actually pop. There you can see them rupturing and releasing their contents. So you can see that this is clearly very different from apoptotic cell death. And so we can now draw this very simple schematic. Receptor activation can trigger caspase-8 activation. This is a depiction of caspase-8 in this dimeric caspase structure, which can lead to apoptosis. But caspase-8 actually also suppresses this alternate form of cell death, programmed necrosis. And this suppression is required for normal development. This is why caspase-8 knockouts die during development. And this is a slightly more complicated version of the same pathway. So you have your receptor activation, which leads to the activation of RIP1. RIP1 activation can then trigger, in some cases, apoptotic cell death via activation of caspase-8, or when caspase-8 is absent, it can lead to the activation of RIP3. This is more or less what I just told you. But of course, this is a very complicated schematic. There's a lot going on here. You have some upstream effects of receptor activation. And we were really interested in studying this, these sorts of phenomena. That is, the activation of RIP3 by the upstream kinase RIP1, and the suppression of these events by caspase-8. And so we wanted to really zero in on the formation of these complexes that lead to the activation of programmed necrosis. And so to do this, to really sort of try to eliminate some of the complexity of this pathway and just focus on this necroptosis-inducing complex, we borrowed a trick from the study of the caspase proteases. And so the caspases, of course, these apoptotic proteases have been around for a long time, and they've been studied for a long time. And one of the strategies that's been used in their study has been this inducible dimerization system. And so what this consists of is the fusion of a dimerization domain, so this protein domain, to the caspase protease, and when you create this chimeric protein with a caspase-8 proteolytic domain fused to this dimerization domain, you can then add this small molecule drug, a dimerizer, which is shown in black here. This brings these two dimerization domains together and leads to dimerization of the protease domains of the caspases, and this then leads to their activation into apoptosis. And this has been well characterized in that context. So we wondered whether we might be able to use this to study directly the activation of the RIP kinases, and in particular, the activation of RIP3. So we fused a dimerization domain to RIP3 and wanted to see whether we could then directly truncate this pathway, eliminate the need for receptor signaling, and directly study how the RIP kinases are activated to trigger necroptosis. So I'm going to now show you a series of experiments employing this strategy. This is a very simple experimental strategy. We create a fusion of RIP3. So here's the RIP3 kinase domain with this FV domain, this is the dimerization domain that I showed you in the last slide, express these chimeric proteins in 3T3 NIH fibroblast cells. This is simply a murine fibroblast cell that importantly doesn't have endogenous RIP3. So the only RIP3 present in these cells is the RIP3 that we add back in. And then we can add AP1, which is what we call our dimerizer, stick these FV domains together and see whether we actually activate RIP3 and what the effects of that are. And I should mention that the structure, sort of the domain layout of RIP3 is such that it has this N-terminal kinase domain, which is shown here in blue, but then it, and very importantly, has this C-terminal domain called a RHIM domain, RIP homotypic interaction motif. And this is a small domain that that's able to mediate the interactions between either multiple molecules of RIP3 or between RIP3 and RIP1. And so these are the interactions that that allow the RIP kinases to interact with each other and that mediate their interactions and the activation of RIP3 by RIP1 during that receptor signaling pathway that I showed you before. And so to measure this, we used the IncuCyte imager, and this is simply a representative movie of some cells undergoing necroptosis. You can see here the counting mask that's put on by the software. These cells again we're using that cell impermeable nuclear dye. So as these cells die, they turn green and they get counted by the software. And this allows us to make these very nice, very kinetically precise movies where we can see cells dying over time at measuring them at very frequent intervals and get these really nice nice traces. And so what we have here, this is just to sort of reorient you in this pathway. We have cells treated with TNF. These are RIP3 expressing cells treated with TNF. You can see that not much happens. But when you treat these cells with TNF, this receptor ligand, in combination with a caspase inhibitor, so remember the caspase-8 suppresses necroptosis in these cells. So when you inhibit caspase-8, you now relieve that suppression and allow this very potent necroptotic response. So this is a RIP3-dependent necroptosis happening in response to TNF plus ZVED. So we then went into our experimental strategy. We took our dimerizable RIP3, we expressed it in these cells, and we induced it to dimerize by addition of increasing concentrations of our dimerizer drug. And what we found was that, in fact, we did get some cell death. We did get some RIP3-dependent death, and this really was due to RIP3. We did several key controls there. We got activation of RIP3 and a sort of modest amount of death, about 30 or 40%. Cell death was as high as we could really get in these cells. Interestingly, we found that this cell death absolutely required the presence of this RHIM domain. And this was a little bit surprising to us because, remember, we're dimerizing RIP3 using this FV domain. And so we expected this to be dependent on this FV domain, but not to actually require this homotypic interaction motif that mediates interactions between different molecules of RIP3. So when we mutated the RHIM domain, we actually completely eliminated the ability of RIP3 dimerization to trigger cell death. And this was associated, the cell death that we saw when we dimerized RIP3 was associated with the formation of RIP3 oligomers, and these oligomers were in fact RHIM dependent. So when we used versions of RIP3 that lacked the RHIM domain, we eliminated the formation of these oligomers. And this is consistent with current models of how RHIM domains and how RIP3 function. So RIP3 has been demonstrated to be to activate by forming these oligomers. And so what we thought we were doing here, this is our sort of our model for what's going on, is that when we dimerize RIP3 in the presence of a RHIM domain, we actually allow RIP3 to then recruit additional molecules of RIP3 via RHIM-RHIM interactions and to form these oligomers of RIP3 that are required for the induction of necroptosis. And so when we mutate the RHIM domain, we now are able to dimerize RIP3, but this is not in fact sufficient to lead to its complete activation. You actually need to form these oligomers to get potent RIP3 activation. So that was all very, very well and good. But what we found, again to our surprise, was that in fact the formation of these oligomers, so just going back a second, so the formation of these oligomers seemed to be modulated by the same enzymes that were modulating TNF-induced necroptosis. So here's TNF receptor-induced necroptosis. When you treat cells again with TNF alone, not much happens. When you add ZVAD, so the caspase inhibitor, to these cells, you get very potent necroptosis. NEC1 is an inhibitor of RIP1. Remember, RIP1 is upstream of RIP3 in this pathway. And again, there's not a lot of cell death with TNF alone, but NEC1 is a suppressor of this pathway. Interestingly, though, when we now treat these cells with dimerizers, so remember this is completely independent of any receptor ligation, this is simply dimerizing RIP3, and we now add ZVAD, we again greatly potentiate this effect. So inhibiting caspase-8 potentiates RIP3 dimerization-induced cell death, while inhibiting RIP1, the upstream activator of RIP3, reduces the induction of RIP3 dimerization-induced cell death. And so this was a little bit surprising to us, right? Here we have activation of RIP3 independent of any receptor, and yet it seems to still be modulated by some of these same players. And we can again look at the oligomerization of RIP3 via Western blot, and also interactors with RIP3. And what we find is that when we dimerize RIP3, in fact, we induce the interaction of RIP3 with RIP1 and caspase-8. So remember, again, these modulators of RIP3 activation in the TNF-driven pathway seem to also be interacting with and modulating the activity of RIP3 when we directly activate RIP3 independent of any receptor. And so what we think is going on here is that that RIP1 must be modulating the formation of these RIP3 oligomers directly. And so what we find is that when we add the inhibitor of RIP1 necrostatin, this leads to a reduction in the formation of these oligomers and in RIP3 dependent cell death. And so, this must mean that RIP1 is directly activating RIP3 in this pathway, right? You put an inhibitor, and the activation is reduced. Well, it turns out that it's actually a little bit more complicated than that, because again, to our surprise, what we found is that when we dimerize RIP3, we get a modest amount of cell death, and when we add this RIP1 inhibitor, that cell death is reduced. But somewhat surprising to us, when we actually knock down RIP1 protein, the cell death induced by RIP3 dimerization is greatly potentiated. Right? So you have opposite effects when you either inhibit RIP1 or when you eliminate RIP1. These respectively block or induce the receptor-independent activation of RIP3. So the presence of RIP1, this is a slightly complicated point, I just want to make it completely. The presence of RIP1 is not required for RIP3 oligomerization and activation. And we hypothesized that necrostatin, this RIP1 inhibitor, may turn RIP1 into an inhibitory dominant negative regulator of oligomerization of RIP3. That is, when RIP1 is present but inhibited, it seems to actually be able to block RIP3 oligomerization. And so this is a sort of a graphical depiction of what we think might be going on. When we dimerize RIP3, this leads to the recruitment of RIP1. This in turn can recruit caspase-8, which is, remember, the suppressor of necroptosis. And so in this context, RIP1 is actually acting in a sort of suppressive role to prevent the oligomerization of RIP3. So given this idea, we hypothesized that in the absence of RIP1 or caspase-8 mediated suppression, RIP3 might undergo spontaneous RHIM-dependent oligomerization in the cytosol. So that is to say, if we, just to step back, if we take away RIP1 and we just look at what happens to RIP3, independent even of RIP3 dimerization, we hypothesize that maybe RIP3 would basically spontaneously oligomerize and form these active complexes. And so to test this idea, we turn to a different experimental strategy, which is this destabilization domain, which this is another protein domain that we can fuse to RIP3. This protein domain leads to the rapid degradation and constitutive degradation of any protein that you stick it on. But when you add this SHIELD1 ligand, you stabilize this domain. And so what you can see here is that this is endogenous RIP3. This is RIP3 with this this destabilization domain fused to it, and it's present at a low level. But when we add our shield drug to stabilize this domain, we induce the accumulation of RIP3 over time. So we can now, independent of any kind of receptor activation or dimerization, we can simply accumulate RIP3 in a cell. And we can now test whether that accumulation of RIP3 is actually being modulated by caspase-8 and RIP1. And what we find is that when we have low levels of RIP3 present, so this is in the absence of the SHIELD1 ligand, low levels of RIP3, and we add TNF, we find that when we knock down caspase-8 in that condition, we get lots of RIP3-dependent cell death. But when we knock down RIP1, we don't, right? So this is consistent with caspase-8 suppressing TNF-driven activation of RIP3, and with RIP1 being required for that process, right? Remember that RIP1 is upstream of RIP3 and is actually a RIP3 activator downstream of the TNF receptor. Interestingly, though, when we now accumulate RIP3 in the cytosol, right? So now we're not activating with any kind of a ligand. We're simply accumulating RIP3. We see that there's a modest amount of spontaneous cell death. But when we knock down RIP1, we now greatly increase the amount of cell death that we see, and this is also the case when we knock down caspase-8. And so what this says is that while in the case of TNF signaling, RIP1 is actually an activator of RIP3, in the absence of that signaling, when we're just looking at spontaneous formation of these complexes in the cytosol, RIP1 is actually suppressing RIP3 activation. And so this was kind of interesting and surprising to us. And of course, we wanted to understand how RIP1 might be able to block RIP3 activation independent of receptor engagement. So in this sort of spontaneous formation of these RIP3 oligomers, how is this being blocked by RIP1? And we hypothesized that RIP1 might cooperate with caspase-8 to directly prevent the formation and propagation of RIP3 oligomers. That is to say, RIP1 is acting at the level of oligomerization to prevent the formation of these RIP3 oligomers that are required for the execution of necroptotic cell death. And so if this were true, we would hypothesize that chemically enforcing RIP3 oligomerization, that is, forcing RIP3 into an oligomer, would remove RIP3 from the control of RIP1 and caspase-8. If we can stick enough RIP3s together using our dimerization system, we might now prevent RIP1 from being able to suppress that event. And so to test this, we added a second dimerization domain, so a second FV domain onto RIP3. These two dual dimerization domains will allow RIP3 molecules to cross-link and actually directly form oligomers independent of any RHIM interactions. And when we do this, we find that we get this very robust, much more robust, in fact, cell death. So here's RIP3 2x FV, so two dimerization domains. We get this very strong response when we add dimerization drug. And interestingly, when we now completely eliminate the RHIM domain, so here we've actually deleted the RHIM domain from RIP3, we can now still get very robust cell death when we induce oligomerization. Now remember that in the dimerization-induced case that I showed you earlier, the RHIM domain was absolutely required, and this we think is because that RHIM domain is needed to propagate those oligomers. Now we've eliminated the need for that RHIM domain by adding this second FV domain so that now we're forcing RIP3 into an oligomeric structure. So as I show here, chemically enforced oligomerization can substitute for RHIM-RHIM interactions. And we can show this very nicely by detecting these oligomers directly. So this is a Western blot following cross-linking. We can see for the formation of these oligomers. RIP3 can still interact with RIP1 and caspase-8. So these oligomers that we're forming are still recruiting RIP1 and caspase-8. But interestingly, now those oligomers are no longer controlled by RIP1 or caspase-8. So when we add the caspase inhibitor ZVAD or the RIP1 inhibitor necrostatin, which remember very potently modulated the cell death in response to RIP3 dimerization, when we've now oligomerized RIP3, we've removed that oligomerization from the control of these players of RIP1 and caspase-8. And so this is consistent with the idea that that RIP1 and caspase-8 are actually acting at the level of RIP3 oligomerization to modulate that function. So this is a model, just a sort of a rough model for the dual function of RIP1 in both activating and inhibiting RIP3. RIP1, of course, as I told you, is required for RIP3 activation during receptor-mediated signaling, so things like TNF receptors activate RIP1. This then activates RIP3 and leads to the formation and propagation of these RIP3 oligomers, which are required for the activation of downstream players like MLKL and the induction of programmed necrosis. However, in the cytosol, we think that RIP3 can spontaneously form oligomers, that essentially RIM domains are a little bit sticky, and sometimes they can stick together and start to oligomerize, and that when this happens, RIP1 is actually also recruited to these growing oligomers in the cytosol, and that this in turn leads to the recruitment and activation of caspase-8, which then suppresses further oligomerization, leads to complex dissociation, degradation, and cell survival. And so in this way, RIP1 can actually both act as an activator of RIP3 in the context of receptor signaling, and as a suppressor of RIP3 activation during this sort of spontaneous cytosolic formation of RIP3 oligomers. And we've now published this study just this year in Cell Death and Differentiation, so if you're interested, you can certainly read more about this work. And I would also just point you to this really nice review that just came out from my friends and former colleagues, Ricardo Weinlich and Doug Green. This is now out in Molecular Cell, and this is a really nice summary of these pathways, as well as a discussion of some of the really beautiful genetic work that Ricardo, Doug, and others have done, demonstrating that in fact all this stuff that I've been telling you actually holds true in vivo. And so finally, I just wanted to of course, acknowledge the people who did the work. This is my group here at the University of Washington. This is Susanna Orozco, who did nearly all the work that I showed you today. I also want to, of course, acknowledge my collaborators, especially Nader Yatim, who I worked with to initially develop some of these systems. He's now in Matt Albert's lab at the Institut Pasteur, and they're close collaborators. Igor Brodsky, Sid Balachandran, and Stephen Tate. And of course, with that, I'd like to thank you for your attention, and I would be happy to answer any questions you might have.
We received a couple questions along the way that maybe you can help us answer. The first, I'm just going to go ahead and paste them in the chat section and read them off as well, and then hopefully you can answer them. So the first question we have is, can you measure caspase-1 activity as a marker of pyroptosis?
Yeah, that's a good question. Let's see. I don't know that there's a great way to measure specifically caspase-1 activity. There are some dyes. I mean, I assume that we're referring to measuring this in the context of something like an incucyte or an imaging setting. There are some dyes, some of the cell player dyes, for example, that have caspase cleavage sites in them, and that become active and that become functional fluorescent dyes only when cleaved by caspases, and those do seem to work relatively well, but I don't know that they're actually specific for caspase-1, unfortunately. I don't think that there are great reagents to differentiate between caspase-1 dependent pyroptosis and caspase-8, 3, and 7 dependent apoptosis. I think that, unfortunately, at the moment, the best way to do that is to use things like knockout technology. So, compare a wild-type cell to a caspase-1 knockout cell and see if there's a difference in its response.
All right, thank you. So there were a couple questions related to your kinetic. You showed some really nice kinetic cell death plots. So a few questions around the dyes and specifically the membrane impermeable dyes that you used. Can you talk a little bit about how you set up those assays and the reagents you used?
Yeah, absolutely, absolutely. So let's go back to this image of dying cells. Um, the way that we've done this throughout these studies is using the IncuSite bioimager, plating a known number of cells, and then adding Cytox green to the media of those cells. And Cytox green, we've tried lots of different red and green dyes. Cytox green is by far the best we've found. It works really well even at very low concentrations. We generally use it in the 25 to 50 nanomolar range, and it works great at those concentrations. So it's also very cost effective because you can really stretch a single tube of it. We add that to the media of our cells. We then treat them however we're going to treat them. And then we simply read out green counts over time, and again, that works really well using the IncuSite. If I can skip forward here to our movie, where did that go? Sorry. Here we go. So this is our sort of our plot of dying cells as measured in the IncuSite using Cytox green. Obviously, what you see here is that what we're measuring is percent cell death, but what we're actually counting in the IncuSite is the number of dead cells per field. And so to get from the number of dead cells per field to percent cell death, we of course need to know our total number of cells, the total cell count, so that we can divide the number of dead cells in each field by that number. And to do that, the way that we found that works best to do that is actually to replicate plate several wells with identical cell numbers to our experimental cells and treat those with cytogreen. So cytogreen, no X on the end, is a cell permeable version of Cytox green that will essentially label every cell in the dish. And so we have actually separate wells that we count using cytogreen that will then allow us to figure out how the percent cell death that we're observing in our experimental wells, which are labeled with Cytox green. And this has worked really well for us. We've tried a couple of other options, but that seems to be the most robust and most cost-effective way to do these types of analyses.
Great, fantastic. So the normalization happens to the cytogreen counts, and the kinetic cell death are the Cytox green counts. Is that correct? Okay. All right. So the third question is coming over now. How do you differentiate necrosis and necroptosis in time-lapse microscopy?
Yeah, that's a good question, and the answer is there's really not a good way. You know, the current status of the field is that necroptosis doesn't really have a good specific assay that differentiates it from other forms of death. And so, you know, in the case of caspase-dependent apoptosis, there are things like tunnel staining and cleaved caspase-3. There are some really nice antibodies that allow, you know, histological analyses or staining analyses that will really help to demonstrate that a cell death event is actually apoptosis. We don't have anything that good for necroptosis yet. So essentially, what we generally do is to measure cell death as defined by loss of membrane integrity, as I've been showing you, and then combine that with things like inhibitors for RIP1 or RIP3, or knockouts for RIP3 or the downstream effector MLKL, and see whether the cell death that we observe goes away in those conditions. And so, you know, we don't have a great sort of single cell analysis tool that lets us really determine that a cell is undergoing necroptosis. Very recently, there was actually an antibody developed to phosphorylated MLKL. MLKL is the downstream effector, again, of necroptosis. That seems to work pretty well in human tissues and in things like histological specimens to define the activation of MLKL, and therefore it should be specific for necroptosis. But that's kind of the best that we have right now.
All right, so a couple questions while you were explaining these kinetic cell death assays that came in. The first being, what cells are we looking at here on slide 17?
These are 3T3 murine fibroblast cells. Sorry, I should have made that more clear. This is a murine fibroblast line that doesn't express endogenous RIP3, but when we introduce our RIP3 constructs, they become very beautifully sensitive to necroptosis.
Okay. And a second question, I think related to this slide as well, comes from Terry. Does fluorescent signal from cell death decrease over long periods as the fluorescent nuclei lift off the dish, i.e. the
Yeah, that's a good question. So actually, one of the great things about the IncuSite for all these assays is that the plates themselves, so the cell dishes don't move, right? So for anyone familiar with the machine itself, you actually put those in a drawer and they stay still, and then the imaging head actually scans around to the different different locations on the dish that you've identified. And so this is different from most like inverted microscope systems where there's actually a moving stage that moves your cells around. And so that's a subtle difference, but it actually is very important for these assays because as those cells move around on the moving microscope stage, they get shaken loose and they drift off into other parts of the dish. And so it makes it very difficult to actually quantify them. Whereas in the IncuSite, there is no movement of the plate itself, and so the cells will actually, generally speaking, stay still and be very cooperative and let you actually image them. This actually even works for imaging of suspension cells. We've had pretty good success getting quantifiable movies of suspension cells using the IncuSite, again, because the dish itself with the cells in it is just completely stationary.
Great, fantastic. One question that came in here from, let's see here, from David. What membrane changes produce the swelling you showed? Water inflow, membrane disruption. I assume that's the Necroptosis movie we looked at.
Yeah, so this is that's a good question, and this is an area of very active research from a number of labs. But so essentially, the current thinking is that that there's this downstream molecule downstream of RIP3 called MLKL. This is a pseudokinase that actually has a very interesting structure. When it's phosphorylated by RIP3, it's been reported to actually directly translocate to membranes and to essentially mediate their permeabilization. It has this membrane permeabilization domain that can directly bind to phospholipids and actually basically rip holes in the membrane and disrupt cellular membranes and lead to swelling and rupture directly. And so, I think that there's understanding exactly how that happens and whether there are other players involved in that process is an ongoing question. But that's the sort of the current thinking downstream of RIP3.
Great. So I think we'll hold on for another minute or two. If anyone has any remaining questions, you can put them into the chat box in the bottom left here. And as we said at the beginning of this, we will be posting this webinar actually to the Essen BioScience website either later this week or early next week. So if you'd like to forward this to colleagues, you can do so at that time. You'll also receive a follow-up email to this webinar shortly. So actually here, a couple more questions here. So Dr. Oberst mentioned about PMLKL antibody a few minutes ago. Was the antibody purchased commercially?
Yeah, so that's a commercially available antibody. I don't actually recall which company sells it, but there was a paper describing it from Xiaodong Wang that I believe was published either this year or late last year that describes the membrane attack by MLKL and also this antibody. And so if you I think it's a molecular cell paper. If you look that up, it'll have all the information, and that antibody is commercially available.
All right. Well, if there are no remaining questions, I think we'll wrap this up. Again, thank you for joining us, and you will receive an email shortly.
All right. Thanks very much.
Yes. Thank you, Dr. Oberst.